Hello students, welcome back to Educators. Today we are with another new video. It's rather a short one on protein absorption. Myself Manjishta Chatterjee. Don't forget to like, share and subscribe. Educators. Yesterday, I've given you a video or uploaded a video on digestion of protein. So, why digestion of protein is very important before just revising digestion of protein is very important before we start with the absorption of protein, because absorption, like in carbohydrate, absorption was only with the monosaccharides, okay, the glucose, the fructose and the galactose. But over here, digestion of proteins does not only include digestion of amino acids but also includes digestion of some peptides, molar peptides, and also rather the polypeptides, some peptones and also proteoses. Okay, so saliva is just for the use of mastication in case of digestion of protein. Then coming to gastric juice, enzyme is pepsin, substrate is protein. We are getting proteoses, peptones and larger polypeptides, which are broken down by the pancreatic juice as the smaller polypeptides and also the amino acid. And also the from the succus entericus we are also getting some amino acids. So all these things, the shorter peptides, the amino acids, they are carried to the villi or the enterocyte for the absorption process. So let us first take an overview of how the protein is being absorbed. So always keep in mind that see this step also, tripeptide and dipeptide. So protein also get absorbed in this form, in some peptide form, and definitely as amino acid. So basically small intestine, mainly the duodenum and the jejunum are the absorptive sites of the protein. Dipeptides, and amino acids, as I've just mentioned, are absorbed. And if there is more than if there is peptides with longer than 3 amino acids, those peptides are difficult to absorb or minimal absorption of such kind of peptides take place. But if it is less than 3, then peptide also gets absorbed. Now some amino acids are absorbed into the bloodstream, but some remains in the enterocyte for the other functions of the cell. And 99% less than of the more than of the protein enters the bloodstream as the amino acid. Okay. So coming to the first type of absorption of protein, which is the peptides. Now, peptides actually, as I've mentioned, that if the peptide is, if a polypeptide is more than 3 or longer than 3 amino acids, then it does not get absorbed. But dipeptide and tripeptide are more rapidly absorbed than the free amino acids. So in case of carbohydrate absorption, glucose were getting absorbed, or the monosaccharides were getting absorbed in all of the places. But in case of protein absorption, dipeptide and tripeptides, they get much better absorbed within the body with the help of this pept1. Okay, pept1, or we can also say that it is a peptide carrier with an active transporter pept1. Okay, active transporter pept1, which is always coupled to sodium and hydrogen. Okay, always coupled to sodium and hydrogen. And they also, as you can see, that hydrogen comes out, sodium gets in, this complex is formed, this coupled reaction takes place, and this pept1, with the help of pept1, the peptide is coming to the enterocyte. Okay, so di and tripeptides are digested into amino acid by the cytoplasmic. So the remaining peptidases, they are digested into the cytoplasmic peptidases. Okay, so the key points of the peptides absorption is, it is the major form where the protein gets absorbed. As the protein gets absorbed, and the rapid absorption is much more than the free amino acid. It is an active transport, or energy is required in this transport. Sodium is also there. Sodium hydrogen exchanger or a coupled reaction is seen, and only free amino acids. So peptides are not getting directly into the blood. Only the free amino acid, they are getting into the blood. Okay. Next is amino acids. There are different 7 types of free amino acid in the brush border, and there are sodium independent characters, which are proton co-transport and facilitated diffusion by this process, and sodium dependent, whereas there are carrier which is coupled to the sodium concentration gradient, as I've just shown you in the previous slide. So over here we can see points are given, like one is dipeptide and tripeptides, amino acids, and larger peptides. Larger peptides are not absorbed, so it is either broken into di and tripeptides. So this larger peptide is broken 80% to di and tripeptides and 20% to this amino acid. Now amino acid moves directly to the blood with the sodium dependent structure, and over here dipeptides, tripeptides, they move for 10%. Okay, so the even some dipeptide tripeptides also, by the reaction as I've mentioned, they also get converted to amino acid. Further it moves as amino acid to the blood, and only 10% of dipeptide and tripeptides move into the blood. Rest of them move to the enterocyte. Now, what is going on within the enterocyte? The basolateral membrane. In the carbohydrate absorption also we have learnt about the basolateral membrane of the enterocyte. So one is the lumen side, the brush border, and the basolateral membrane, the opposite side of the enterocyte. They are actually exporting the amino acid, the basolateral membrane, with the help of the sodium transporter, with the movement of, as I've mentioned, that it is an active thing. So ATP utilization is there. Along with that, there is a sodium potassium movement is there. So diffusion and also sodium dependent independent carriers are also there, and free amino acid, as I've told you several times in this video, that free amino acid are absorbed directly into the blood. So protein transportation, or protein can be either absorbed as peptides, dipeptides, or tripeptides, but it is much faster than the amino acid. They move into the enterocyte. After that, they are changed into amino acid, and finally with the help of this sodium dependent transporter, and it is by an active movement or sometimes by diffusion process, 90% of the amino acid move to the blood, and 10% of the dipeptides, tripeptides move to the blood. So I hope you have understood the digestion of protein. It's a simple, short and crisp absorption. I'm sorry. Uh, Short, crisp absorption process of protein. That's all for today. Tomorrow I'll be coming with another video, that is digestion of fat. So stay tuned with educators. Thank you.